Right now, somewhere inside a womb, a cluster of cells is spinning. Not metaphorically. Actually spinning, like tiny propellers, and the direction they spin in is about to decide which side your heart ends up on, which side your liver ends up on, and scientists now believe whether you will spend your entire life reaching for a doorknob with your right hand or your left. For 50 years, geneticists thought handedness came down to a single switch. One gene flipped one way or the other before you were even born. They were wrong. And the real answer, the one hiding in the DNA of over 1 million people, connects your hand to something far stranger than a coin flip. It connects it to the same ancient molecular machinery that decides which side of your chest your heart beats on. Stay with this one, because by the end, you will never look at your own hand the same way again. This is not internet speculation. This is published, peer-reviewed genetics coming out of some of the most respected labs in the world. The Max Planck Institute for Psycholinguistics in Nijmegen, where geneticist Clyde Franks has spent close to 2 decades chasing the molecular roots of hand preference. The University of Oxford, where researchers combed through brain scans looking for the physical fingerprint of laterality, and a 2020 study published in Nature Human Behavior that pulled together genetic data from the UK Biobank, from 23andMe, and from research consortiums around the world into the largest handedness study ever conducted. This is not a fringe theory. This is mainstream science quietly rewriting one of its own oldest assumptions. To understand why that matters, you need to understand what scientists used to believe, and the analogy is simple. Imagine you are trying to figure out why some houses on a street have their front door on the left and some have it on the right. The old theory said there was one master switch somewhere in the neighborhood's blueprint, a single decision point, and every house just inherited a copy of whichever version was passed down. Find that one switch, the thinking went, and you have solved handedness. That idea has a name. It is called the right shift theory, proposed by British psychologist Marion Annette back in the 1970s, and for decades it shaped how an entire field thought about the problem. Annette's theory proposed a single gene, sometimes called the right shift factor, that a version of that gene left the outcome more open to chance. It was elegant, it was simple, and for a long time it was the closest thing the field had to an accepted explanation. In 2013, a research team led by William Brandler, working with Oxford's Clyde Franks, thought they might have actually found it, a gene called PCSK6 on chromosome 15, linked to relative hand skill in people with reading difficulties. Headlines ran with it. A single gene for handedness, finally located. Here's the part that changed everything. It didn't replicate. When larger, more rigorous genome-wide studies went looking for PCSK6 fingerprint in the general population, it wasn't there. Not a trace. The single gene model, the one that had anchored the field for 40 years, quietly collapsed under the weight of better data. And what took its place wasn't a tidier answer. It was a far messier, far more interesting one. In 2019, Franks, working alongside geneticist Dorrit Boomsma and colleague Caroline D. Koval, published a study in Scientific Reports that went back through the molecular evidence and effectively closed the door on the single gene idea for good. Hand preference, they concluded, was not one switch. It was hundreds of switches, each one contributing an almost imperceptible nudge, stacked on top of each other like grains of sand until they tipped a scale one way or the other. Geneticists call this a polygenic trait, and the analogy here is worth sitting with for a second. Think of it less like a single light switch and more like a dimmer with 1,000 tiny sliders, none of which does much alone, but which together shape the final setting. Then came the number that changed the scale of the entire investigation. In 2020, Gabriel Cuellar-Partida and a sprawling international team published a Genome-Wide Association Study, a GWAS, which works by scanning the entire genetic code of enormous numbers of people at once, looking for spots where left-handed people and right-handed people consistently differ, the same way you might scan every house on every street in an entire country looking for the handful that were built with a different blueprint. Their sample size was staggering: 1,766,671 people. To put that in perspective, that is roughly the population of a mid-sized country like Guyana or the Bahamas many times over, or, closer to home, more people than could fill every seat in every NFL stadium in America, combined several times over. And out of all of them, the team identified 41 distinct locations in the genome tied to left-handedness, and 7 more tied to ambidexterity. 48 locations, not one gene. 48. Sit with that number for a second, because it reframes the entire question. For half a century, the field had been searching for a needle in a haystack, one gene among 20,000, assuming that once it was found, the mystery would be closed. What Cuellar-Partida's team found instead was that the haystack itself was the answer. Handedness wasn't hiding in one needle. It was distributed in tiny fractions across dozens of separate threads woven through the genome. None of them decisive on its own, all of them pulling in the same rough direction. Tissue enrichment analysis, a method that checks which parts of the body a set of genes tends to be most active in, pointed squarely at the central nervous system. This wasn't noise scattered randomly across the genome. It was a coordinated signal, concentrated almost entirely in the tissue that builds the brain. That number alone should have been the headline. But buried inside the data was something even stranger, something that pointed toward an answer nobody had been expecting. If you've made it this far, you already know this channel doesn't do surface-level science, so if you want the rest of this reveal to land the way it's supposed to, hit subscribe now, because what comes next connects your hand to your heart in a way that sounds almost too strange to be real. So, quick recap before we go further. Scientists spent 50 years hunting for a single handedness gene. They thought they found it in 2013. It didn't hold up. By 2020, the real picture had emerged. 48 separate genetic locations spread across the genome, each playing a tiny part. But when the Cuellar-Partida team looked closer at what those 48 locations had in common, one category of genes kept showing up again and again. Genes involved in building something called microtubules. Here's where the analogy earns its keep. Every cell in your body has an internal skeleton, a scaffolding of hollow protein tubes called microtubules. And if you want a picture in your head, think of the steel framework that goes up before a skyscraper gets its walls. The cranes, the girders, the internal structure that everything else gets built around. Microtubules do that job inside individual cells. They guide growing nerve fibers to where they need to go. They form the internal machinery that lets a cell divide correctly. And critically, they build tiny hair, like structures called cilia, which some cells use almost like oars to move fluid or to move themselves. The Genome-Wide study kept flagging microtubule genes near the DNA regions tied to handedness. That was strange enough on its own. Then, in 2024, a team led by Dick Shiven, alongside Sorena Sohaili-Nezed, Simon Fisher, and once again Clyde Franks, published a follow-up study in Nature. Communications that went hunting not for common genetic variants shared by millions of people, but for rare ones, the kind of DNA misspellings that show up in only a small slice of the population. Working with exome data, the protein coding portion of the genome, from over 38,000 left-handed people and more than 313,000 right-handed people drawn from the UK Biobank, they found something that stood out immediately. A gene called TUB4B, a beta-tubulin gene, literally one of the building blocks of those microtubule scaffolds. Rare protein-altering variants in TUB4B showed up 2.7 times more often in left-handed people than in right-handed people. 2.7 times. That is not statistical noise. And 2 of those variants were frameshift mutations, the kind of genetic error that doesn't just tweak a protein, it can derail the instructions for building it entirely, leading the cell to discard the transcript altogether. Something was breaking, subtly, in the scaffolding of the brain's earliest architecture in a way that nudged a person's dominant hand. What makes this so significant is that TUB4B wasn't even one of the tubulin genes flagged back in the 2020 common variant study. It was a completely separate signal, found using a completely different method, looking at rare mutations instead of common ones, and it still landed in the same biological neighborhood. In genetics, when 2 independent studies, using 2 different techniques on 2 different slices of the population, both arrive at the same category of gene that is not a coincidence researchers wave off, that is triangulation. That is the kind of convergence that turns a hypothesis into something closer to an established finding. A separate research review published in Trends in Genetics in 2025 by Jichang Shah and colleagues pulled together this entire body of evidence and confirmed the pattern. Across nearly every major handedness study of the last decade, tubulin genes and the microtubule pathway kept resurfacing. Again and again, like a signature the genome couldn't quite hide. Here's the part that connects it to something much bigger than handedness. Microtubules and cilia don't just live in brain cells. They also live in a tiny structure in the developing embryo called the embryonic node, and around 3 weeks after conception, before your brain has even begun to take shape, cells in that node use cilia to physically spin fluid in one consistent direction. That spin is what tells your body which side to put your heart on, which side gets the larger lobe of your liver, which side your stomach curves toward. It is the literal physical origin point of left-right asymmetry in a human body. When those cilia fail to spin correctly, a rare condition called situs inversus can occur, where a person's internal organs form as a mirror image of the usual layout, heart on the right instead of the left. Researchers have traced disruptions like this to genes such as FOXJ1 and CATNB1, both of which are deeply tied to cilia and microtubule function, the very same molecular family the handedness studies keep circling back to. Nobody is claiming that the gene that puts your heart on the left is the same gene that makes you write with your right hand. To be clear, the picture is more layered than that. But the biological toolkit, the microtubule and cilia machinery that builds physical asymmetry into your organs in the first weeks of life, appears to be part of the same broader system that later helps sculpt asymmetry into the two hemispheres of your brain. Your hand preference may be a distant echo of the exact same ancient cellular process that decided, in secret, weeks before you had a face, which side of you would be which. Think about the scale of what that implies for a moment, because it deserves two comparisons, not one. On one hand, picture the embryonic node itself, a structure so small it would fit many times over inside the period at the end of this sentence, active for a matter of days before it disappears entirely. On the other hand, picture the outcome it sets in motion, a decision that echoes for an entire human lifespan, 80, 90 years of reaching for things with the same hand, tens of millions of individual motor choices, all downstream of a spin that happened in a structure smaller than a grain of salt for less than a week, before you were even the size of a grape. Let's pause and restack what we know so far, because this is the moment it all clicks into place. Scientists spent decades looking for one gene and found 48 genetic regions instead. Those regions kept pointing to microtubules, the cell's internal scaffolding. A 2024 study zeroed in on a specific tubulin, gene TUB4B, with rare mutations nearly 3 times more common in left-handed people. And that same tubulin, cilia-driven machinery, is the literal system responsible for deciding which side of your body your heart forms on. In other words, the same ancient biological toolkit that lays out the architecture of your internal organs may also be quietly nudging which hand you reach for a cup with 20, 30, 60 years later. In the next part of this, it's worth going straight into what this convergence actually means for the brain itself, because the story doesn't stop at organ placement. There's also a question worth sitting with here, and it's one that researchers themselves have raised. If handedness and brain asymmetry share a genetic root system, what else grows out of that same root? Scientists, including Sebastian Aucklenberg and Onur Gunturkun, whose work on brain lateralization spans decades, have shown that the same hemispheric specialization tied to handedness is also tied to how language gets organized in the brain, and a 2019 study out of Oxford, led by Akira Weiberg, found overlapping genetic signals between handedness and neurodevelopmental conditions. The Cuellar Partida team's own 2020 data found a positive genetic correlation between left-handedness and both schizophrenia and bipolar disorder. Not a cause, not a diagnosis, but a shared thread in the underlying genetic architecture, the same way two different fabrics might be woven from strands of the same dye-lot. That overlap doesn't mean being left-handed says anything about a person's mental health. What it does suggest is that the developmental machinery building brain asymmetry is wired into more of your neurology than anyone expected when this research began. Now, for the part that has to be said plainly, because getting this wrong would misrepresent the science entirely. None of this means genetics fully determines whether you're left-handed or right-handed. Twin studies, going back decades and repeated as recently as 2022 and 2023 by researchers including Lena Pfeiffer and colleagues, consistently show that heritability for handedness sits at roughly 25%. That leaves the majority of the variation unexplained by DNA at all, shaped instead by factors in the womb during development, and possibly by simple biological noise in how a growing brain settles into its final asymmetric structure. Identical twins, who share effectively the same genome, do not always share the same dominant hand, and that single fact alone tells you genetics is a strong contributor here, not a verdict. Even studies of twins raised entirely apart, separated at birth and reunited decades later, show the same pattern. Shared DNA nudges the odds, but it does not lock in the outcome. There is no individual test today that can look at someone's DNA and predict with certainty which hand they will favor. What these studies show is population-level pattern, not individual-level prophecy. The 48 loci, the TUB4B variants, none of it functions like a single dial you could turn to flip a person's handedness. It is closer to weather than to a light switch. Many small forces interacting, producing a trend that is real but never fully predictable in any one case. And it's worth remembering that roughly 90% of people across every culture and every era, researchers have been able to study, land on the right hand as dominant, while left-handedness holds steady at around 10% worldwide. That ratio itself has barely moved across generations, which tells its own story. Whatever the system is, it is old, it is stable, and it was already fully in place long before anyone was around to study it. It's also worth being honest that this field has been burned before. The PCSK6 story from 2013 is a reminder that a single promising result, even a published peer-reviewed one, can dissolve once a bigger, better-powered study comes along. Other early candidate genes met the same fate. LRRTM1 and SCTDB2 both generated headlines in their moment. Both looked promising in smaller studies, and neither one held up once the larger genome-wide datasets arrived. Scientists like Clyde Franks and Simon Fisher have been careful, in their own writing, to frame TUB-B4B and the microtubule pathway as a strong lead, not a finished answer. Replication is still ongoing, sample sizes are still growing, and the honest expectation across the field is that this picture will keep sharpening, not settle overnight. That caution is not a weakness in the research. It is exactly what separates durable science from the kind of single study headline that gets forgotten a year later. And if this kind of story is the reason you're here, the kind where a decades-old scientific assumption gets quietly dismantled by better data, that's exactly what this channel exists for. So make sure you're subscribed before the next one drops, because the next thread we pull on goes even deeper into the DNA than this. So where does that leave the coin flip you were born with? Somewhere around 3 weeks old, before you had eyes, before you had a name, a handful of cells in a microscopic structure inside your mother's body spun fluid in one direction, and in doing so, quietly began sketching the blueprint of your entire physical asymmetry: heart, liver, stomach, and, it now appears, the deep neurological wiring that would one day decide which hand reaches first for a pen, a cup, a doorknob. You never made that decision. No one did. It was made by a spinning fringe of cilia, executing instructions written in genes like TUB4B, genes scientists spent 50 years failing to find because they were looking for one answer instead of 48. The next time someone asks you why you're left-handed or why your brother is right-handed despite having grown up in the exact same house, you can tell them the truth. The decision was made before either of you had a face, by microscopic machinery neither of you will ever consciously feel, echoing forward from the earliest architecture of the body into a habit repeated 1,000 times a day for the rest of your life. Tell me in the comments which part stuck with you more, the idea that your handedness might trace back to the same system that shaped your heart, or the fact that scientists chased a single handedness gene for 50 years before admitting there were 48 pieces to the puzzle all along.